Hello everyone. Very good evening to all. Am I audible? Please confirm. In your chat box, if you could quickly confirm if I'm audible to you and you're able to hear me clearly. Hello, Yash. Welcome to the session. Thanks, Suman sir, to teach this us for free. Of course, yes, Biotechnica is, that's what Biotechnica is for. We try to help the student community as much as possible. And our CEO MD, which is Shekhar Suman sir, this is his visionary that we are going to influence one student at a time. And thank you so much for joining. And definitely, you are the future of life science. Definitely. We would love to see you shine one day. All right, so I can see many participants joining in here. So you have heard about AI/ML in bioinformatics, AI/ML in biology, AI/ML in drug discovery. You all have learned so much from all these courses of Biotechnica. And here we are. Yes, we heard you. We took your suggestions, and here we are with a completely new domain that it's not new, but yes, Biotechnica has brought to you something which we have not done before, and we thought that why not, our students should be enlightened about this. And today we are here with a very in demand topic, hot topic for pharmacists and other chemistry background students. So this is for you. Every time in all our webinars we will be getting that when can we have something regarding chemistry, can we have something AI/ML applications in chemistry, cheminformatics. So here we are. Once again, a very good evening, everyone, and a warm welcome to all of you, the bright young minds who have joined us today. And I'm Urmimala. I'm delighted to host this exciting webinar, that is AI plus Cheminformatics. It's the future career formula for drug discovery and pharma innovation. Now, let's start with a simple thought. What if molecules could think? What if a computer could predict whether a drug will cure a disease even before it's synthesized in lab? So that's not fiction anymore. That's what AI-powered cheminformatics is, and it's redefining the way we discover new medicines. And in today's world, so much hype is there for artificial intelligence, machine learning. It's no longer optional skills for you. They consider them kind of a lab equipment of modern biosciences. So whether it is for predicting protein structures or to design new molecules, AI is turning data into discoveries faster than ever before. And the truth is, researchers who can bridge biology with computation, they are the ones in demand, and they are the ones who can shape the next generation of pharmaceutical innovation. That's exactly what today's session is about, how AI and cheminformatics together are transforming drug discovery, and most importantly, how you can build a career at the intersection of science and technology. So please help me welcome our esteemed eminent speakers for the evening. Our first speaker for the day is Dr. Tiliban Manivarma. I welcome him for the session, and thank you so much, sir, for joining us today. So, Dr. Thiliban Manivarma holds a PhD in computational biophysics from Nicholas Copernicus University in Torun, Poland. He is deeply passionate about leveraging cutting edge technologies in computer aided drug design, computational chemistry, and machine learning to advance drug discovery and structural biology. His expertise spans biomolecular analysis, ligand modeling, and virtual screening, all aimed at accelerating innovation in therapeutic development. Dr. Mani Verma will open the session with predictive chemistry, where algorithms meet molecules, helping us understand how AI predicts molecular properties long before even experiments begin. It's a pleasure to have you here, sir, and we are looking forward to your session. Thank you for joining.
Thank you so much for the introduction, Urmimala. And let me share my screen. And
If we could have the introduction of the speakers, and then we will move forward to the first to your session. Yes, thank you, sir.
yes, yeah.
We have Dr. Snigdha Tiwari, ma'am. She is a computational biologist and clinical research specialist at Biotechnica CRO and a PhD from IIT Roorkee. She has worked extensively in protein ligand modeling and bioinformatics, and even co-developed the Utsarjan app, a digital health platform for pediatric nephrotic syndrome in collaboration with AIMS Delhi. And ma'am will talk about the industry shift, how pharma is investing in AI, and what it means for you. Okay, that's going to be a very important session. You should know what all is going on in the real world. So expect real world insights on how top pharma companies like AstraZeneca, Takeda, and Eli Lilly are building entire AI divisions and what kind of professionals they are hiring. That's the most important thing, right? Finally, we will hear from Dr. Shubhi Singh, an interdisciplinary researcher with a PhD in biotechnology from SRM Institute of Science and Technology. Her expertise spans molecular docking, ADMET prediction, and bioinformatics workflows for target identification. Dr. Singh will guide us through becoming an AI-ready chemist, skill sets and tools. You should know what all you need to know for becoming an expert in this field. And ma'am will share what it takes to transition from wet lab to data lab science and how to master the AI tools driving the future of molecular discovery. Thank you so much, Snigdha ma'am and Shubhi ma'am, for joining us. And our audience is looking forward, along with me, to your sessions. Yeah.
Thank you, ma'am. Thank you so much for introducing us.
Yeah, my pleasure. So we will definitely, good heart, we will be taking up the questions at the end. So I will request everyone to grab your note copy and pen, because coming ahead is a lot of informations for you. All our speakers are going to talk about many important things, and you should take a note of this. And you can type your questions in the comment section, drop your questions below, and we will be having a Q&A session later, where we will be picking up your questions and our esteemed speakers will be addressing them. Okay, so without much further delay, let's begin with our session for the day. I hand over the controls to Dr. Mani Verma. Sir, if you could please start with your session. Sir, you are on mute.
Yeah, I'm sorry. And I'm sharing my screen.
Yes.
Let me check. Can you see my presentation? If one could confirm me.
Yes, sir. If you could please make it full screen.
Yeah, can you see the screen
Yes, sir.
now?
If you
Super.
could change the next slide.
Okay, fine.
Is it going towards next slide? Yes, sir. It's
It is
good.
fine. It's
Yeah,
changing,
it's fine,
right?
sir.
Super,
Yes, sir. It's
super.
changing.
Yeah,
Thank you.
yeah. So, hello everyone. My name is Thiliban Manivarma. So today I'm going to be talking about the topic on the predictive chemistry, where algorithms meet molecules. And before I'm stepping into my slides, I would like to mention that all the pictures and memes included in the slides with respect to the, which goes to the credits to the respective, the creators in the social media and the research article and most of the images are picked up from the Google Images. And yes, so the first slide, which will be describing about the traditional drug discovery, you know that it's really slow and costly. And you can see this is one of my favorite images. You can see this is a drug discovery pipeline, starting from the scratch. Once you get the disease, what you want to find the drug. So we have to understand the disease studies such as structural information of the target and all the sequence analysis and all this information should be done in the beginning. So you can see it is costly and it's also time consuming. Starting from the scratch to the end, you can see it might take minimum 10 plus years for the drug to come to the market. And also if some of the drugs if it get fails at the end of the phase, such as like clinical trials, it needs to be coming from the scratch again. So it's a vigorous process, I must say. So we need to understand how we fasten this particular process by the machine learning and AI. And yes, you can see all the stages like disease diagnosis, target identification, lead optimization, and preclinical studies, all of them where we can use this AI ML to fasten the process. And yeah, so yeah, this is the image which explains the evolution of AI in drug discovery. So in 1990s, this was the first one of the breakthroughs in computational drug discovery, where they found the SMILES notation. SMILES notation is nothing but the simplified molecular input line entry system, so which describes how the molecule is looking like. You can't feed this drug structures to the machine learning model. Machine learning model won't understand the structure. So we need to represent in a way which is a series of strings, which from the strings we can get the fingerprints, which is a binary vector like zeros and ones. So computer and algorithms only understand the numbers. So we need to make the molecule and we have to convert the molecule to the numbers. So such a way we make the machine learning model or computer's algorithm to understand the drug structures. Then in 2000, they find something called like quantitative structure activity relationship, which is a statistical modeling where we get the data from the experiments. So we build the model using linear regression, logistic regression. From that, we try to predict the activity and toxicity, different property of the drugs, which was in like 2000s. But we are currently in the AI era, which is 2010s. So from that here, we are here in the AI revolution. We have like AlphaFold, structure prediction, and like generative AI using reinforcement learning. A lot of technologies are coming up. By the way, this image was created by Google Gemini. I directly thanks to it. And yeah, so before I'm stepping into the real world applications of machine learning for drug discovery, I would like to give some simple example, like a funny example to understand for the people who don't have this background of machine learning. So let's take this one of the South Indian dishes called dosa and idli. If the people here, if you don't know like what they are, this is the South Indian dishes. It's a pancake. The dosa is a pancake, and idli is a steamed cake of rice. So I'm taking this as an example to describe what are the different types of machine learning we have. And firstly, we have the supervised machine learning. You know, the dosa is looking like a golden brown color, and the idli is a white color. So by looking at these images, we can easily say, okay, this is dosa, this is idli. So we can easily classify the data by the labels, right? If the data is having the label, when you are applying this data set to the machine learning model, it's a supervised learning. So when you have the data with the labels, and it's a supervised learning. Whereas you can see in unsupervised learning, there are different types of the dosa. Uh, you can't group them by looking at the eyes based on the taste, right? You eat them based on the flavor, you can describe them. Okay, this is spicy dosa, this is sweet dosa, this is chocolate dosa, this is veg dosa, non-veg dosa. So you can classify them based on the taste, right? So you don't know what the label is, but based on the the characteristics, you are grouping them. So this is called unsupervised learning. Whereas in semi-supervised learning, we have both labeled data and unlabeled data, and using both of them to use this machine learning model is called semi-supervised learning. Whereas in reinforcement learning, let's say you are trying to make a dosa with the help of your mom or dad. So we as a kid, we are trying to get the information from our parents, like mom and dad, they are telling, hey, you should make a dosa like this in the round shape. Where every time you make a dosa, initially it gets bad, right? So they are the feedback system. They are giving you, okay, this is -1, this is not good, this is not in a proper shape. So eventually you will learn from them. Okay, every time you do, you learn, you do mistakes, you learn from them. So -1, they are giving you some score. So eventually when you're doing great, they are giving you positive score. So you are learning from the mistakes with the help of the feedback system. This is called reinforcement learning. This reinforcement learning is helping in the generative AI. People are using this technique a lot. And yeah, so again, one of the examples is supervised machine learning is that we as a kid, when you are trying to walk, first, like initially your parents, like mom and dad, they will tell, oh, you should walk like this and come. Okay, so the way we get the information from them, we try to learn, and after some point, and you start learning, right? So now you are walking like a boss. And second of all, because this is supervised learning, because you get all the information from them, they are telling, okay, you should walk like this, you should not walk like this, when you fall you have to get up. They are guiding you with the labels. So this is called supervised learning. Whereas unsupervised learning, when I started cooking, so I try to cut the vegetables, I try to group them in a one place. So meat alone and vegetables alone, spices alone. So I'm trying to group them based on the characteristics. So I'm grouping them. This is called supervised, unsupervised machine learning. So the final product is here. So the reason that I'm giving you all of this example is to get you into the world of machine learning. So these are the basic concepts that we have. And again, in machine learning, some of the famous concepts and the techniques we should know is features and labels. These are the two fundamental concepts in machine learning. One is features, so which describes what the data is. Let's say we take idli and dosa. We are trying to classify, right? So the idli is characterized by the different features such as shape, color, texture, thickness, and cooking method, right? And this is called features. The features are the input variables or attributes that are used to make the predictions or classify the data in machine learning models. They are measurable properties. We are giving the properties, attributes of the data, right? So it's also known as predictors, input variables, independent variables, attributes, and covariates. The reason that we are calling independent variables is that all the features are independent. So the dosa, it needs not to be with the same size. Look, sometimes you can see the triangle dosa. So all the features are independent, but the labels, all the labels, which is let's say dosa, all the features, thin, orange, crispy, thin, and the steaming, all these features are depending on this dosa. Dosa is depending on all these features, right? So this is the reason it's called dependent variable, and whereas features are independent variables. So these are the two techniques we should understand here. So the overall steps in machine learning is we get the data. You can get the data from the public data sets such as Kaggle and PubChem for drug discovery. And you can either do it by Python web scraping to get the data from there, or you can use the SQL, the programming language, to extract the data from there. And once you get the data, you have to make the data is clean and neat. So where we have this data pre-processing by different Python packages, Panda, NumPy, SciPy. And once you get the model, you need to know what kind of problems that you are facing, whether it is supervised problem, unsupervised learning, or semi-supervised. Based on the data, we are choosing the problems and what model that you want to try, which is also important. So you can do it by different packages in Python, such as scikit-learn, keras, and TensorFlow. And finally, you are evaluating the model. using different matrices. So I'm not going in depth to all these like properties, but I'm just giving you an overall introduction to the machine learning here. So code can't smell a molecule. So whatever the images that you are feeding, like as I said initially, the machines, they don't understand the structures. So we have to convert the structures or images. Let's say you are working with the cancer, like the tumor data, the cancer data with the cancer images. So machine learning, they won't understand. So we have to convert all these images to the numbers. You can see when you're zooming the images, you get the different pixels. So each and pixels represent the numbers right there. So whereas here you have the structure, you get the smiles form, the string form that will tell you what are the smiles looking like. So you get all this information from the smiles, and from the smiles you get the fingerprints, you get the all numbers from this data structure and this drug structure. And these are the widely used data in the drug discovery, especially in small molecules and biomolecules. So let's take this let's take the small molecule. You can see this is the structure. So this structure, computers and machine learning won't understand. Machine learning model won't understand. So we need to get the smiles notation. So each and every drug structure or chemical structures, you are having their own smiles form. So from the smiles form, you can get the molecular fingerprint. There are different molecular fingerprints such as like MACCS Max fingerprint, Morgan fingerprints. So in the future, if you guys are having any doubt, I will happy to discuss with you all, but I'm not going in depth to that. But the overall idea is we need to convert the strings to the numbers. So let's take, you have the protein ligand data. You people might have done docking, right, to the interaction. So when you have this protein ligand data, protein data, you can make those structures into the graph form. So graph is nothing but series of nodes is connecting by the edges. You can get all the features from the graph, the node features and edge features. So those represent the numbers. So you can see these and all like representation as numbers, so which this machine learning and AI model will understand. So if you are working with the sequence, like the protein sequence or genomic sequence, there's something called one hot encoding. So which is looking like this, like in a matrix, you have binary data like zeros and ones. So from this, the machine learning model will understand. But recently we are in AI era, the deep learning era. So you don't need to convert all the strings or the structures to the numeric form, like in a numeric form. There is advanced AI algorithms called recurrent neural network and convolutional neural network. They are specially made for the images, the CNN, convolutional neural network, which is specifically made for the images. So you don't need to convert the images to the particular numbers. So it automatically gathers information. It scans each and every pixels. It does automatic innovation. Whereas this recurrent neural network, when you are passing all the smiles, it automatically understands, okay, these are the sequence which will one after one, so I will understand all of them. So this is the AI era. And some of the applications with respect to the drug discovery, I'm going to be explaining. So the first is like toxicity classification. So which flags whether the molecule is having the toxic or non-toxic based on the experimental data. So you can get the data from your colleagues who are working in the lab, like in the experimentalist, such as like medicinal chemist, organic chemist, and synthetic chemist, like whatever the people they are working there. So reactivity prediction also you can do, whether the molecule will cause a reaction or not. And bioactivity, we have like different like quantitative and structure activity relationship, like different absorption, distribution, metabolism, like, all these properties which can be found with the help of this AI and ML. And like one of the simple case studies, this quantitative structure activity relationship, which finds the particular molecules, what it exhibits, like high C50 inhibitory constant 50, like HEC50, that excitatory concentration, and lethal dose 50. This is a toxic dose calculation. All the details, like you get all this information from the databases such as Kimball, Zinc, Binding DB, Drug Bank. From all the sources, you can get the data. Once you get the data, you can build a model. It can be either classic machine learning model or deep learning model, such as like artificial neural network, convolutional neural network, or this AI, like a reinforcement learning. It depends on what the problem that you're working with. So molecular structure data, biological activity data, pharmacokinetic data, all this data that it can be found online there. So all these databases you can see here. And yeah, so same as like toxicity prediction, the databases such as ToxCast and Tox21, and which is having multiple data set. And yeah, so we use different algorithms to build the model. So yeah, so this is one of the case studies they mentioned, like generative models like variational autoencoders and generative adversarial neural network, which is also finding the which is helping us to find this toxicity of the molecules. And yeah, so this absorption, distribution, metabolism, excretion, all these properties, all the data that you can find it online from the database, and from this we build the model and we can get the job done using multiple applications. So since my talk is like with restricted to 15 minutes, I can't explain all this the practical hands-on at this moment. In the future, if the things are going fine, we can have this short session, how can we implement right there. And these are one of the famous packages, open source packages, which is totally free. If you have a laptop, if you have this Python installed, you can use all of them. So one of them is RDKit, which is a Swiss Army knife, I must say, which is handling all kind of properties, data processing, file processing, fingerprint generation, the descriptor calculation, everything in one place. You can do all these properties prediction. I mean, like the processing, which can be done by this RDKit, and something called DeepChem, which is kind of advanced packages, which is having all this AI and ML dependent applications and processing for both drug discovery and material science. And yeah, most of us, and we were working with the structures of the biomolecules, so which is done by AlphaFold. So right now we have AlphaFold 2, 3. It's coming up. Things are going fine for the people who are don't have the structures of the target. We have this AlphaFold to get the job done. And yeah, so the conclusion is that we have this AI which is analyzing the data behind the human capability. Anyway, like let's say you have like millions and billions of data, you can't analyze everything from the scratch, right? It's a hectic, vigorous process. So I must say, so using this AI, and we can try to understand the data much better. We can find the patterns inside and patterns hidden in the data set. And yeah, so faster innovation. So with the help of this AI/ML, we fail fast. So you can generate like 50 ligands or 50 compounds. You can easily find whether it will be active or inactive. Once you find it inactive, you can come back to the starting stage. So without wasting time. So fail fast, iterate fast is the motivation behind this AI for drug discovery. So of course, we are stepping towards this AI, which is supporting the greener science. It reduces the lab waste, right? Let's say you are synthesizing like 100 compounds in the lab. So it's a cost. You are spending a lot of money for each and every raw materials for synthesis. At the end, if you find it is inactive, that's it. You are gone. The money is gone. So instead of like spending time on synthesis for each and every compounds, we use AI to get the top 10 compounds. Okay, if you are confident enough on the 10 compounds, you go for the synthesis. If not, you come back. So fail fast, iterate fast is the motivation behind this AI for drug discovery. And I tried my best to explain you a simple understanding of this machine learning. And these are some of my favorite memes. I find it online. So deep learning is nothing but advanced ML, machine learning model. So it's not like you are learning everything from the deep, from the swimming pool. It's not like unsupervised learning without any guidance. okay, this dog is rocking fast. And yeah, some of the, because machine learning is all about data. If you are feeding the wrong data, you get the wrong calculations. So you see there it's trying to classify the dogs and cookies. Some of the cookies are looking like a dog, but anyway, if the data is wrong, everything is wrong. So if you I tried my best to make you understand like what is there behind the scenes of this machine learning and AI for drug discovery. So if you have any queries from my talk, feel free to reach out to me and we can have a discussion. So you can ask any questions in the end. I'll be here. So yeah, so I really thankful to the host people, like they invited me for the talk, and I tried my best to explain you people. And yeah, so I'm stopping my screen.
Absolutely, sir.
Yeah.
Yeah, absolutely, sir. It was really a fascinating session, I must say, and the insights that you showed us, how algorithms are reshaping molecular discovery, and your use of real life examples was amazing, and it explained the complex concepts like supervised, unsupervised learning. It made it so informative and so easy to understand, and it was wonderful listening to your session. Time just flew by without us even realizing it. And definitely, sir, this was not enough. Listening to you, so we would love to have you again in one of Biotechnica's future sessions or workshops, if you could share more of your experience and expertise in this field. So thank you once again for the talk, sir. We will have
Thanks.
you again question answer session at the end. Yeah. Okay, so students, if you have any questions for Dr. Mani Verma, please feel free to post it in the comment section, and we will definitely pick it up during our Q&A session. Moving forward to our second session now, I invite Dr. Snigdha Tiwari ma'am to kindly take the controls and conduct your session, ma'am.
Thank you so much, ma'am. Good evening, everyone. Hi. So today I'll be dealing with how the industry shift is happening, guys. Okay, like how the industry is shifting. The pharma companies are shifting towards AI and ML. You have seen how AI and ML is playing big role in in the chemistry, like in the docking and QSAR studies. You can convert your chemical formats into like fingerprints and do it with a very little time. So that is the pickup for today. And I'll be dealing with how the industry shift is happening, right? I mean, how pharma is investing now in AI and ML field, and what it means for you. Like, how you can pick up the opportunity. You can now pick up the opportunity in growing AI and ML field, right? So I'll be also talking about the real world insights into the AI adoption, industry trends, and also the new career opportunities for all of you who's listening to me. Um, Talking about why AI is reshaping pharma, right? I mean, why this shift is really happening. What AI and ML is doing, like with chemistry. So first of all, guys, the pharmaceutical industry is at the digital turning point. As Sir explained it very well, that gone are the days when when we used to put a lot of cost and time in clinical research, then drug repurposing, or like the trials and everything, and then it gets failed, right. I mean, at the end, the hypothesis is rejected. I mean, the statistics are skewed. So that is the real catch here. The pharmaceutical industry is actually now shifting towards AI and ML, the big data analysis that we say. Now things are shifting to the SQL, to Python, to making your chemicals into into fingerprints. Then Sir very well explained, you need to do you have to pick up your niche now, for for selecting AI in like all this era of AI and ML, right? So AI and data science now drive every stage, right? From drug discovery to clinical trials and then patient outcomes. Like if it is a drug discovery, whether it is your clinical research and trials, whether it is a drug repurposing QSAR studies. AI and ML is now driven in the chemistry, as we say, the cheminformatics, the word, if we can explain that way. The key drivers are exploding the omics data, right? How genomics, proteomics, and metabolomics is actually exploding with AI and ML, the big data analysis. Rising the R&D cost, of course, with the big data, with, because things are going global. In the research and in the clinical research and trial, as we talk about, we need to include we are going we are going into the world, right? I mean, we want data sets and population and samples from all over the world. So that is, that may rise the R&D cost. So need for faster and precision based drug discovery. So now is the time that we need a precision based drug discovery. And that is why AI is actually reshaping now. Scale of AI investment in pharma is huge, guys. You can see how, AI investment growth in pharma industry is rising, comparing from 2019 to 2023 and like 2025 now. Picked up this data from global AI in pharma markets. I mean, the vlogs, the web, the global AI in pharma markets is actually increased so much, for $1 billion, like more than that in 2023. So projected to reach about $11 billion by 2028, and CAGR, like if we talk about, the business point of view, to 23% and plus. The top spenders that you can note down, and you have a spot on, you can just create your space there, if you have all the niche and all the data set and skill set that we talk about. AstraZeneca, Pfizer, Novartis, Takeda, GSK, Sanofi, these are one of the top spenders in, AI in pharma. So major investment areas that they are doing, and, the AI assisted target identification, drug design and molecular screening, predictive toxicology, and clinical trial optimization. So each of it sir talked about, right? I mean, how to get your hands on the toxicology, how to, know about the QSAR, how to use RDKit, supervised, unsupervised learning, making out the fingerprints, and then, loading it, with the encodings and everything. I mean, you have to select your niche, and then, develop the skill set to, to have your spot in, in the upcoming, industry. AI plus CRISPR, this is the smarter immunology therapy that, that we are doing now. People are using CRISPR as, as an intelligent tool. People, as I say, that, that of course the industry and, the pharma companies and the industry, $555 million partnership in 2024 to integrate AI driven CRISPR screening for immune and inflammatory diseases, right? So in the immunology, in the CRISPR screening, picked up from the Financial Times 2024. I've just, I've just put in, I'm just showing you screens, with the sources, like, the Financial Times and all of that. I've just put the sources also to let you know that how much, you have the opportunity, how much you have the opportunity in the trending of AI today. I mean, you just have to, make yourself equipped and make yourself, make your skills solid, right? So Algen uses AI to identify genetic targets from CRISPR libraries. AstraZeneca is one of it, applying the findings to next-gen immunology pipelines. The impact that they are putting in is, the faster validation of the drug targets and reduction in trial and error research, right? I mean, of course, if you're using AI, if you're using CRISPR, if you're, if we are using all those that Sir explained, I mean, the supervised learning, machine learning, all of that, it will reduce the trial and error in the research. It will, we'll get the faster validations of the drug targets, right? This is how, the smarter therapies, machine learning, artificial intelligence are coming in. So building AI leadership roles, right? I mean, this is the spot for you. So how will, whoever's listening to me must be thinking that, how to get a spot, right? I mean, so building AI leadership role is, you can have a look that Takeda is, one of a biopharma hiring associate directors, cheminformaticians in 2025 postings. Also the roles they're inviting, all the, this is just one of the, pharma that I've put in, there are a lot who's started their AI and ML modifications in 2025 posting. So the role they are offering is AI and ML driven molecule design, then multi-omics integration, the collaborations with computational and medicinal chemist. The key insights to for you to to watch on is the traditional roles like medicinal chemist are evolving into like AI chemist and chemoinformatics data scientist. So now you'll be watching the job postings like the data scientist, the chemoinformatics data scientist, or AI chemist. Gone are the days when the people, or the AI industries, pharma industries, everyone was hiring about the traditional roles, right? I mean, they're not really sticking to the traditional roles now. This was the source from Takeda Career Portal of 2025. I've just taken the information from there. Talking about the global hub for AI talent, right? Talking about the hub globally for artificial intelligence and the talents out there. 1,500 AI and digital experts to be hired on, like 2026. If we talk about 2026, things are just moving forward. You can see the graph, it's just rising. And till 2026, everything is all the AI digital experts. If you talk about the pharmas, the startups, uh, they're just eyeing on hiring like about 1,500 and big roles. Like, I mean, it will go beyond, I'm sure. So focus areas, what are the focus areas? And that you have to note it down. Predictive analytics for clinical data, automation and trial designs, AI powered patient recruitments, right? If you talk about the clinical research, clinical trials, drug designing, repurposing, repositioning, everything is has moved on to the AI based. There was the time when clinical research, the attrition rate, everything was on the papers, right? You can you distribute papers, you make questionnaires, but now it is just out of focus. Now you need to have the hands-on on the SQL, Python, NoSQL databases to to collect your data and then do your machine learning models on it, right? So a strategic location in India, if I talk about, then access to computational and clinical expertise is important. Initially, before the introduction, I'm told that it is not just someone's wanting. It's not just someone is choosing. It's a need now, right? I mean, it is a dire need for everyone to to select their niche and start working on AI and ML. If right, if you want to work in the intersection of chemistry, artificial intelligence, machine learning. The source that I picked up is the Times of India, 2024. Talking about global hiring and job market trends, right? What is the global hiring, that is going ahead, and what is a job market trend right now, guys? So 41 year-on-year rise in AI and ML jobs in India, life sciences sector. So picked up from the Economic Times 2024. Growing demand across roles are computational biology, AI for drug discovery, clinical data scientist, like science, and bioinformatics and chemoinformatics. So bioinformatics, chemoinformatics, drug discovery with AI, computational biology, clinical data scientist, clinical research with with the expertise of Python, MongoDB, SQL, NoSQL, all of it is actually rising, and it will rise in the coming years. India is also, India is a big market. India is offering a big, big market, because it is a big market as the epidemiology, as a as a population, as a sample size. So uh, I mean, of course, a great deal to to work within India or globally, right? Emerging career titles. This is important. I know what is the the career titles that is emerging now. Traditional roles were there, with medicinal chemist, bioinformatician, clinical analyst, pharmacologist. These were the traditional roles. But then, the evolved roles today, with the intersection of artificial intelligence and machine learning, are these: the AI chemist, the cheminformatics data scientist, the digital health data scientist, and computational biologist. So everything has moved. Can you just compare the traditional role keywords and the evolved role keywords? The keywords will only tell you that how AI, ML, chemoinformatics data scientist, computational biologists. So computational data, AI and ML, everything is just evolving around them, right? Whether it is a chemistry, whether it is drug discovery, whether it is an omics data, or whether it is a clinical research nowadays, right? The core skills that you need, that you need for entering in any of the intersections, like entering into any of the like any of the uh, you know any of the spots that you pick up, is the core skills. Sir also explained it very well, that you should have hands on Python, your RStudio, SQL, NoSQL databases, TensorFlow, RDKit. RDKit, these are the libraries that will help you making the fingerprints. The TensorFlow, machine learning, deep learning models, scikit-learn, data visualization, few of the libraries like Plotly, Power BI. Then bioinformatics, right? I mean, this is bioinformatics is a very basic and very important, of course, to get started with the insight of how databases work, how primary, secondary, composite databases work for you, right? I mean, picking up a data is important. How will you make the knowledge graph? How will you make establish your databases? Or how can you go ahead with the libraries like TensorFlow and RDKit? So you need to first start with the databases, right? So that is so important. Getting equipped with NCBI, Uniprot, PDB, PubChem is are the basics. Very important one. Then AIML for prediction, clustering, imaging, and sequence analysis. Sir has already explained, right, how clustering, prediction, image analysis, and sequence analysis, one can do, with deep learning, with the different libraries, machine learning, and so on. Talking about the salary and the global demand insights, few of the salary and the global demand insights that I've just you know picked up from ZipRecruiter, the LinkedIn salary insights 2025. The latest one I've just picked up are, if you're joining as an AI chemist in India, you can spot on 12 to 18 LPAs, and then you can change it to dollars if you're going globally, like in the USD setups. Then come the Cheminformatics scientists, all kind of scientists. The Cheminformatics one, maybe the data scientist, 10 to 12, computational chemist 8 to 15, and AI clinical analyst as about 7 to 14. I mean, this is the salary bars and setups in India, and then global also USD is just put up the conversions. You can note it down. These are the salaries and global demand insights that that one can look into, and get an idea of how much AI and pharma is actually spending. What it means for you guys. I mean, whatever I've shown onto the screen still now, what that means to you, what that means for you. I mean, how you can go ahead with with the intersections of different with the different topics that we all are talking about. So work at the intersection of AI, biology, and chemistry. So that means for you, how you can start working onto the intersection of artificial intelligence, machine learning, knowing about biology, knowing about the databases, bioinformatics, and of course chemistry. Join global research collaborations, that is so important. How you can get ahead with joining global research collaborations. There are a lot of them. You can publish AI driven discoveries in top journals. Starting if you're starting your your journey in, whether it's an academia or transitioning into the industry, start with the AI driven discovery. Start reading first with what is going ahead, and how your niche is actually making sense with what you're reading, of course. And then you can start it with with writing into the journals, making how the manuscript, and then of course targeting into the top journals. Upskill for high growth digital pharma roles. Of course, that is the main thing. You have to first grow yourself, right? I mean, you have to upskill your your digital pharma roles. You have to upskill your all that matters, right? That is, of course, that is important. Roadmap to enter the field. What is the roadmap that you can start and enter to the to all this field? Phase one is learn the foundation, most basic and of course mandatory. First, you need to have skills. First, you have to, you need to just have patience and build your foundations very strong with Python, R, statistics, and biology. Right. Explore the applied AI tools. You have to, whatever AI tools and the the work packages, the libraries, You can explore all of them. Few of them explained by Sir. Just put up some of them like RDKit, DeepChem, BioPython, PyTorch. These are few of the deep learning and supervised unsupervised modeling libraries, right? I mean, RDKit will help you with the fingerprints. BioPython will work you with all the omics related, bio related setups, then all of that. Then hands-on mini projects. Once you set up, once you select your niche, once you learn a good foundation based on your niche, and then explore the applied AI tools, then you go to phase 3, that is hands-on mini project. You start building mini projects for yourself, right? Docking automation, or a drug target prediction, or maybe virtual screening, or QSAR modeling. Start making yourself a mini projects. Start making yourself a GitHub repository, so that whenever you go ahead for for picking up any spot, you can just show it to them. You can just show your peer groups about what you're doing. So hands-on mini project is very important. The network and collaborate, at the end, that works. I mean, you can start your journey with with Kaggle, with LinkedIn's different hackathons, your GitHub repositories. You can show up, you can share in your GitHub repository to your peer, to your to your scientific peers, right? So that is how the roadmap to enter the field works, right? I mean, end to end. So this is thanks a lot for listening, and being active here. Thank you so much. So this was all that I wanted to tell you with how to enter the roadmap. For all the upskilling, you can you can join different, different platforms that that we are providing, or anyone else is providing, of course. Joining the collaborations, joining the listening to the webinars and everything. Just upskill yourself all the time. So thanks a lot. Over to you, ma'am.
Yes, thank you so much, Dr. Smigda ma'am. It was very eye-opening your session on how pharma industry is actually what picture you showed us, the real picture of how this pharma industry is embracing the AI. And it was very necessary for our audience, not just learning the skills, but where it can be utilized, and what are the stepping stones, the milestones that they have to follow. It was very helpful, and I'm sure our audience now sees how and where the real and exciting opportunities lie, and what opportunities are there, that and what they need to do to grab hold of them. So thank you once again, ma'am, for your session. We will take up the questions at the end in Q&A session. Okay. Okay, so with that, we are now going to have the third session. I invite Dr. Shubhi ma'am to kindly share the screen, and please start your presentation. And so I have a question from Yash. You're asking that is it beneficial for me or from third year BSc chemistry? I referred this from Rasayanika. So as you can see on our slides also, and even in this YouTube, like the brochure flyer, what you're seeing in front of you, we can see Rasayanika is also a part of this because it is chemistry related, and that is why Biotechnic and Rasayanika in collaboration is bringing to you this particular session and this particular subject, okay, where you can definitely learn a lot. Okay, so I think, Yash, that clarifies your question, and we will take up more topic related questions at the end. Yes. Shubhi ma'am, over to you.
Yeah, thank you so much, ma'am. Is my screen visible?
Yes, ma'am, it's visible.
Yeah, okay. Thank you, ma'am, for confirming. So yeah, hello, good evening, everyone. So myself, Shubhi. So in the previous two sessions, you guys have come across like what are the job opportunities are there in the AI field, like AI in chemoinformatics field. If you become the chemoinformatician, okay, you have learned about, like in the first session, as sir have told, you have learned about the various AI methods, like machine learning method, like supervised, unsupervised, and how it works in a very good way. You have been learned, like till now, the basics of AI and ML, and the what are the outcome you are going to get after this course. Okay, so here in my session, I'll be talking about becoming an AI ready chemist. Okay, skills and the tool sets. Okay, so what are the skills and the tools you need to know to become an AI AIML chemo in chemoinformatics. Okay, so here I'll be talking about what you need to know, what are the skills you should have, what are the tools you should be aware of to step into this field. Okay, yeah. So yes, first I'll be talking about what is AI and ML in chemoinformatics. Okay, so what is AI and ML in chemoinformatics? So here, as we all know, as we have seen in the previous session, the AI stands for the artificial intelligence. Okay, so it basically enables the computer to think and learn. So with the help of an AI and with the help of ML, we can allow the computer to think on behalf of human. Okay, so here we are not like instead of humans, here the computer will work for you. Okay, it will predict the model, it will train the model, it will find the different patterns from the chemical data. With the help of machine learning, we can find the different patterns from the chemical data, we can work with that. Okay, so it will make our work easier. Here we don't need too much of labor to do it. Okay, so with the help of AI and ML, we can narrow down the we can do the work in the shorter time and in a proper way. But the only thumb rule for the AI and ML working with it is the data which we are giving as an input for the AI model to learn, that should be very accurate. It should not have like it should not be a false data or like the or not the good quality data. The main thing which we need to learn, like keep it in our mind, the data which we are going to give over and which we are going to feed to our system, it should be a proper and the accurate data. Okay, so yeah, so we will be looking at the various applications of like AI and ML in the field of chemoinformatics. So first I have like it predicts the molecular property. Okay, so as sir have told about the different molecular properties, like we can say like the like molecular weight. Okay, like hydrogen bond donor, hydrogen bond acceptor, what could be the log p value, that is like the molecule could be hydrophobic or the hydrophilic or the lipophilic, what it could be. Okay, so with the help of AI and ML, we can predict the properties of the molecule, like how many donors are there in the molecule. Okay, like what is like the in the chemistry we have like the different NH groups are there, alkyl groups are there, so we can get to know all these things with the molecular properties. Okay, so next application which we have it over here is it optimize the reaction condition. Okay, so with the help of the AI and ML, we like as we perform all these things at the wet lab level and at the lab level. Okay, with the AI and ML, we can optimize all these things at the using the machine learning methods. Okay, using the computer itself, we can optimize a different condition. Okay, fine, based on the properties of the molecule, we can say like, okay, fine, this way this reaction will work out, this is the particular condition of this particular molecule. Okay, so with the help of that, we can figure out that. Next, we have the designing a new compound. Okay, so with the help of the QSAR, like as sir has also mentioned in the about the QSAR, and even the Snigdha ma'am have also mentioned about the QSAR, that is a quantitative structure activity relationship. Okay, so it is basically help for the prediction of the new compound. Okay, so it will helpful for us for the prediction of the new compound with the existing data. How the QSAR works, basically with the existing data, the machine will learn and it will produce the new compound with the various like properties of it. Okay, various molecular properties or the biological properties of it, and then we can take those particular compound which we have got it from the QSAR, and then we can work on it to the wet lab, or we can perform the molecular docking study, simulation studies, and we can go ahead for the drug discovery process. Right. So next, with the help of AI and ML, we can have the automated drug discovery pipeline. Okay, so with the like as in the first session, you must have seen the whole drug discovery pipeline sir have mentioned, right? Starting from the target identification, lead optimization, and then it's like validation, target selection, each and everything you must have seen in the first session. Okay, so it's just so what just happened here, instead of doing the each and every step individually, we can automate the pipeline itself with the help of the AI. Okay, so we can automate the whole pipeline of the drug discovery with the help of the AI and ML. Okay, so next we'll be talking about how AI and ML is transforming in the research pipeline. Okay, so as we all know, the traditional drug discovery method, it's a very time consuming method and it's a very costly method. Okay, why? Because like in the golden time, what researchers or the scientists, what they used to do, they used to take up, suppose you have like 100 compounds, they used to check each and every compound like at the wet lab level one by one, and then because of that it used to take a lot of time for it used to take up a lot of time for the researchers, and it used to be a very expensive technique. Okay, so all these things used to take up a lot of time to come up with a new drug in the market. But nowadays, like it as we all know, the drug discovery process, it probably take like 10 to 15 years for the one drug to come into the market. Okay, so these are like, so that's why we say the traditional discovery method is like a long it's a very long process and it's a costly process. But nowadays we can make with the help of AI, we can make our work very much easier. Okay, so now we have like the AI driven workflow, and that makes our work faster and predictive. So what we do, we download, like suppose you say the millions of compound from the different databases like Duke's database or the Impact database. Okay, these are some databases that is like from the that have the natural compounds. Okay, that is derived from the plant source. So we download it and then we just directly use the AI and ML. Okay, with the help of it, like, and we need a very high computing system for this to perform, and then we can that makes our work very faster. And and that, as I told you in the first slide itself, in the AI and ML, the data with which the machine is learning, that should be very accurate. Okay, so that should not be a false data, the data should be very accurate. So that makes our work faster and the more predictive. Like we can predict like the more precise work we can do with this. Okay, so like here I have given you the pipeline. Okay, so pipeline of, suppose, like the drug discovery with the help of like AI and ML. Okay, so first we take up the data. So data, like what here, like suppose we have like some biological data, like either from the some different databases, or maybe like some of your colleague or your researcher who have identified the new compound from the some plant source. Okay, we can take that biological data, then we convert those into the descriptors. Okay, so as sir mentioned that the machine learning, we cannot give the biological format. So here we have to convert those biological format into the descriptors, that is the binary number we have to convert it into. Okay, then we perform the QSAR model. Okay, we, with the help of QSAR, we predict the like the IC50 value, EC50 value, and then it's like we predict the molecular weight, what is like each and every property of the particular compound. Okay, with the help of QSAR, then we go for the docking studies. Okay, so now our compound is ready, and then we want to check this particular compound against the target. Okay, so suppose one gene or the protein or the enzyme is responsible for causing disease. Okay, so we want to suppress that particular protein with the help of the compounds which we have identified. So we go for the protein ligand study, that is the docking studies. Then we check the admit prediction. Okay, so admit stands for the absorption. Okay, absorption, metabolism, excretion, toxicity. Okay, so here what we do, like we look for the admit properties of it. Suppose like the hydrogen bond should be like acceptor should be less than this. So there is a different rules is there for the admit prediction. So we predict based on the admit property, we predict our compound. Okay, if the compound passes the admit property, then we send this particular compound for the validation. Okay, so this is the end step of the drug discovery pipeline, what we do it in the chemoinformatics background. Okay, so here we we integrate this with the cloud and the robotic and the automated synthesis. Okay, so we integrate all these things with the help of the cloud, machine learning models and the AI ML model. We use it with the help there. We use like the different cloud servers or the robotics or the different automated synthesis which is like already available. Okay, so that's what we do with that. Next, we'll be looking like the key computational technique ever chemist should know. Okay, what are the computational techniques the chemist should know to step into the AI and ML field in the chemoinformatics background? Okay, so first step we have it is like the data pre-processing. Okay, so what is data pre-processing? So data pre-processing is basically once we get the data after your sequencing, like your genomic, like your whole genome sequencing or your transcriptomics or like your metagenomics, any of the sequencing or the proteomics. Once we get the data, we need to clean the data and standardize the molecular data. So we need to clean, like we need to remove the errors and every other thing from the data. Then we need to convert that particular data into the descriptors, that is the numeric representation of the chemical structure. So we need to convert all these things into the descriptors. Then only our machine learning model will understand the biological data or the or like the chemical data whatever you are giving as an input. Okay, next we need to train the model. Okay, so we have like a different classification method and the regression method. Okay, and different using the different ml using the different ml algorithm. What we do, we train the data using the regression model or the classification model. We train the model based on these. Okay, then we go for the after the model is been trained, it has been trained, then we go for the molecular docking analysis. So here what we do, we predict the ligand target binding. Okay, so as I have mentioned, like we will have like the receptor or the target, that is the known compound, that is a known protein, gene, or maybe the enzyme. Okay, and we use the predicted compound against this target. Okay, and then we try to figure out the binding score of it. Okay, next after we predict the binding score of the protein target, okay, like a protein and ligand, then we go for the simulation and validation. So simulation and validation, why do we perform? To get to know the accuracy of the protein ligand complex. Okay, so the complex which we have got it after the molecular docking, based on the binding score, we can get to know the more accuracy, like how accurate the model the how accurate the protein ligand complex is. With the help of the simulation we get to know, and we look the poses of the protein and ligand in the every nanosecond, what is happening and how the poses of the protein and the protein and the molecule is changing. Okay, so here it's mentioned like refining prediction through MD. Okay, so molecular dynamics simulation. Next, we will be looking for the molecular representations. Okay, so now as we have talked about the descriptors and every other thing. Okay, so now there are different types of representation of molecules are there. So first we have like the smiles format. Okay, so as we go to the PubChem database, like PubChem is a database where like the natural compound and the synthetic compound both will be available. From there you can download the compounds. Okay, so SMILE stands for the simplified molecular input line entry system. Okay, so this is a very common format which we found in the PubChem, and then we download this format from the PubChem, that is for the compound, and then we then we then we convert that into like the PDB format for our further molecular docking and other work. Okay, but usually the molecules will be present in the smile in the databases in the smiles format. Okay, next we have like suppose your smiles format will be like the suppose you can take like C double bond O, like it will be in the chemical formula. Okay, it will be written as a chemical formula. Okay, like how we have it in the chemistry, how we write the chemical formula, the same way it will be written in the smiles format also. Next, we have the descriptors. Okay, so descriptors, what we get to know here, we can get to know like the physicochemical property or the topological property or the electronic feature. Okay, we look for all these things. We change the like the different the biological entities into the binary numbers. Okay, with the help of the descriptors. And then we have the fingerprintings. Oh, so fingerprintings are basically again the binary pattern. So in this we use like Morgan and MACCS, as sir have mentioned about these two terms earlier. Okay, then at the end we have like the graph based representation. So okay, so graph based representation, what's this happen? So we show like in the graphical format. Okay, and then where in the graphical format, the each atom will denote as a nodes, and each bond will be denoted as the H. Okay, in the molecular representation for the AI and ML and the chemoinformatics. Okay, yeah. So next we have the AI and ML algorithms for the QSAR and the and property prediction. So as QSAR, we have, like I have mentioned about this earlier, so basically we have like the different models that can be used for the prediction of the QSAR and the properties of the compound or the molecule. Okay, so we have like the regression model. There we have like linear, SVR, random forest, XG boost. Okay, so these are some of the regression model which we use it for the prediction of the property. Next we have the classification model. There we have like SVM, decision tree, or the neural nets. Okay, so here we can create the, it will be looking like a phylogenetic kind of tree which we can create for this in the classification model. Then we will have like a deep learning model. Okay, so there we have like the graphical neural network or CNNs. Okay, so here we have like the different types of model. And then we can use all these models to predict like the solubility or the toxicity or the IC50 value, EC50 value. Okay, or like the pharmacokinetics of the molecule. So we can predict using all these models. We can predict the different properties of the compound that we have derived from the, that we have got it from the QSAR or the already exist, that suppose somebody have identified in the wet lab level and we don't know how it can be identified. So we can use all these AI ML algorithms, and then we can identify the properties of it. Okay, next we have the base, on the basis of the molecular docking and the structure based modeling. Okay, so here, so predict how the molecule binds to the protein. Okay, so molecular docking is basically for the predicting how the molecule, suppose we have a molecule and this is the protein, so how the one molecule can go and bind to the protein. Okay, so this is what is called as your docking. So, okay, so how this can be performed at the structure based modeling. So first in the docking workflow, if I tell you, the first step is the ligand and the protein preparation. Okay, so what just happened, the ligand and the protein. So ligand, as I mentioned, it will if you go to the PubChem database or the any other database, mostly it will be available in the SMILES format. Okay, so that SMILES format we convert into the PDB QT format. Okay, and even the protein, which protein we downloaded basically from the PDB. Okay, that is a protein data bank. So from there, if we download the protein, it will be available in the PDB format. Okay, then so what we do, we convert the ligand and the protein into the PDB QT format, because the docking platforms like your Autodoc or the Schrodinger, whatever the platform tools you are using, that's Autodoc's mainly it uses PDB QT form. Okay, so next we have the defined binding site. So once our protein and the ligand is ready, it is prepared, then we define the binding site where the ligand should go and bind to the protein. Okay, so we give the particular grid box kind of thing, and then we say that okay, at this particular place only the protein, the ligand should go and bind to the protein. Next, after the grid box been designed, then we perform the docking. Okay, and then we get the score for the different poses. Okay, so suppose in the Autodoc Vina, like we get the 9 different poses. Okay, so 9 different poses will have the 9 different like the binding energy score. Okay, so we will get the different binding energy scores in that. Next, we there what just after that, the PDB, the after the performing the docking, the output file will be generated, and that output file we can either visualize in the PyMol or in the like PLIP soft tool. Okay, we have the different analyzer tool, or in the Chimera we can go there and we can visualize the interaction, like the different hydrogen bonds between the ligand and the molecule, what are the amino acids are interacting with the protein. Okay, we can get to know all these things with the analysis. Okay, so here I have mentioned few tools like Autodoc Vina, Pyrex, Schrodinger, Gold. These are few of the tools that can be used for the molecular docking or the structure based modeling. Okay, yeah, so next we'll be looking for AI-based protein and ligand analysis tool. So what are the AI-based protein and the ligand-based analysis tool? So first we have like the AlphaFold and ESMFold. So as sir have also mentioned about this, the AlphaFold, it predicts the 3D structure of the protein, and I hope like you all must be aware of this term, because it was like this term was already in the limelight, and then everybody was keep using, all the chemists and like all the scientists, when if they want to step into the bioinformatics, everybody was using the AlphaFold, and it's a very easily used tool. It's like is anybody can use it, even a naive person can use the AlphaFold. Okay, so it's like it is used basically for the prediction of the 3D protein structure. Next we have the DeepChem. Okay, so this is the ML toolkit for the chemistry. So okay, so in this like all the it was based on the ML algorithm DeepChem. Then we have the ChemProp and the RDKit. Okay, so ChemProp, these two are used for the for the molecular data pre-processing and the modeling. Okay, so like with the help of the RDKit, we can convert the smiles format into like the fingerprints and the descriptors, and we can, with the help of it, we can get to know like the various properties in every other thing. We can use RDKit for that. Okay, next we have the Molbert and the GraphCon. Okay, so these are basically the molecular feature extraction using the deep learning model. Okay, so RDKit also is a machine learning algorithm, whereas these two are the other two that can be used by the uses the deep learning for the feature extraction of the compound or the ligand or the molecule. Okay, so now here we come to now we have understood what are the basic like things, what are the tools which are there, the AI based or the ML based, what are the models which is there in the AI and ML based. Okay, through this till now. Okay, now we'll be looking at the core skill demands. What are the core skill demands you are there in the job market, or either in the like the projects if you are going to do. Okay, so what are the core skills that the companies and the every any other startups or anybody is asking for nowadays from the chemoinformatics. Okay, so you should have basically the technical skill. You should know like Python, RDKits, skikit-learn, PyTorch. Okay, then other thing you should know about the QSAR modeling, molecular docking, MD simulation, right? We have seen all these things. And then for the data visualization, as Sneha ma'am has also mentioned, that for the data visualization, we have like the Matplotlib, Seaborn, all these few technical skills you should be aware before stepping into the job market, because the job is needing the candidates who are like in a like very good in all these skills. Okay, then you should have like a conceptual knowledge also along with the technical skill. So you need to be having like statistical reasoning, then chemical intuition, then understanding the model interpretability. Okay, so you need to be aware of all these things. Okay, so these are few of the technical skill and the conceptual skills which I have mentioned it over here, right? So you are supposed to know all these things before you step into the job market. Next, from wet lab to data lab science. Okay, now we till now we were working with only with the wet lab. We used to do each and every experiment, and then we it used to take up a lot of time. It used to consume a lot of money. At the end, sometimes we don't even used to have a proper outcome out of it. Okay, now what we are doing from wet lab, we move to the dry lab. That is the data lab science. We have moved to the dry lab. So now we are adopting to the programming mindset, that is your Python, R, C, C++. These are the few of the languages we are learning, programming languages. Then start small. So what it is, so it's basically the automated data analysis. Okay, we are creating an automated pipeline that is helping us to do the automated data analysis. Okay, now we are collaborating basically the computers and the experimental data. Okay, now with the help of an AI and ML, we take up the experimental data and we work further with the computational data. We convert that data into the computational part, and we use AI and ML, and we generate the output out of it. Okay, next we have the interpret data, like we need to know the statistical analysis or the chemical reasoning. All these things we are supposed to know while we are working with the AI and ML also. Okay, because statistics and every other these things can be used at the machine learning and in the dry lab also. Okay, it's not only for the wet lab. We use all this analysis for the dry lab experiments also. Okay, so for upskilling your for your upskilling, you can opt for the online courses, like how the Biotechnica is going to offer you the AI and ML AI and ML chemoinformatics. Okay, so soon we are going to start up with this tool, and they will be talking about the DeepChem and the different whatever the bioinformatics tools which I have mentioned over here. Okay, what are the AI and ML based tools which I have different models, regression model, whatever I have been mentioned. We'll be exploring all these tools and software and the different kits during the AI and ML chemoinformatics tool, which we are going to start soon. Okay, so there you will be learning about more in detail regarding the how the AI and ML is stepping into the field of the chemoinformatics, okay, in the field of the chemistry, and how we are converting the chemistry also, we are connecting chemistry also with the AI and ML. Okay, yeah, so here we have the future of molecular discovery. So now we have like the generative AI models, so that designs the novel molecule. Okay, then we have like the digital twins. Here we it's like the it simulates basically the molecular behavior virtually. Okay, then we have like the autonomous labs, like here we have like AI guided synthesis and the testing of the compound. Okay, we have every other thing. Then we have like the different ethics and the interpretability of the data. So AI is basically an assistant, not the replacement of the humans. Okay, so AI can never replace the humans. Okay, so we always have to keep that in mind. We need AI to walk with the world. Okay, we to be with the world, we need AI, but it will never replace the humans. Okay, yeah, so for AI also we need humans to feed the data and do every other things at the background, right? Yeah, so here I am winding up my session. So thank you very much, and thank you everyone for listening to me. I hope you like this session. Thank you all.
Yes, thank you so much, Shubhi ma'am, for that inspiring session. And I'm sure our audience have benefited. And it's very much important to know the skill sets specifically. And thank you for the wonderful talk on the skill sets and the tools which are needed to become AI ready. It was a very practical and a motivating session for all our inspiring researchers here. So once again, I extend my heartfelt thanks to all the amazing speakers for such insightful and thought-provoking sessions. And in short, today's big takeaway is that the future of drug discovery belongs to those who can speak the language of both chemistry and computation. AI is not replacing scientists, it is empowering them to think, design, and innovate faster than ever before. Now, before we move on to our Q&A session, I would like to share something truly valuable for those who want to take their learning from today forward into real life hands-on experience. So we have our Biotechnica's AI/ML in Cheminformatics course. Our next batch starts from 28th October, and this program is designed to help you gain practical, industry ready expertise in AI/ML in drug discovery. And you will work directly with all the cutting edge tools which you just heard from our eminent speakers like DeepChem, RDKit, DevTalk, AlphaFold, Haddock, all guided by expert mentors who are there on the screen. So whether you choose a 3, 6, or 12-month project along with the course, you will also earn a certification which is backed up by hands-on research experience in case you're going for the projects also, personalized mentoring, and even placement assistance, and even some resume building support as well. So if today's session has inspired you, then this course is your next step towards becoming that rare professional who understands both molecules and machine learning. Believe me, students, this is the perfect blend that pharma companies worldwide are searching right now. So do not be delayed in this learning process and do not feel left behind, right? So a quick note, our seats are limited. So if you're really serious about transforming your research career, then this is the moment to take that leap. Okay. All right. So now let's open the floor for our Q&A session. I invite all our speakers on the stage, and I have got a few questions from our attendees, and our speakers will be happy to share more insights and guide you on your journey into AI-powered drug discovery. We will not be able to take up all questions, but certainly we will take up a few questions. And in case you want to connect with our speakers later, you can do so. I think Dr. Mani Verma has already shared his contact details, his social media platform details, and Dr. Snigdha ma'am and Dr. Shubhi ma'am, you can contact through Biotechnica. Okay, so we will just pick up a few questions from here. So my first question is directed towards Dr. Mani Verma, and this is from our audience, Pallavi. She wants to know how is Cheminformatics helping in optimizing reaction conditions or predicting synthesis routes. If you could throw some light on this, sir. The question is on the screen.
Yes, yes. Yeah. Yeah, there are some reactions. And I'm not sure because I haven't had any uh, experience working with the reaction conditions, but there are some databases like Reaxys, and where we could get to know about like what are the different functional groups and what are the functional groups which are causing this toxic and non-toxic reactions. And based on the such information, we feed all this data to the model to make it understanding. So also predicting the synthetic synthesis route. And the second part, which I'm not sure, so I can't answer and I can't answer that. But yeah, so based on the information that we get it from the Reaxys database and other information, we try to build the model to uh, for the for such applications, I must say.
Okay, okay. Thank you, sir. Thank you for the insight. So moving on to our next question. This is from you. Currently, most industries recruit a computational chemist with PhD. So is that true or not? I think the student or the participant wants to know something about this. Dr. Snigdha, ma'am, any insights from your end regarding this?
Yes, ma'am. Hi, you. So, if you you can, what you can do for that matter is, you can reframe your niche. I mean, you can reframe your niche accordingly. You can you can start your journey with the analyst, with the with the data scientist, AI and ML, maybe interns in drug discovery, or like Cheminformatics project rules. So build your practicals, build your foundation solid, and then you can maybe collaborate or volunteer in different research projects. If you don't want to go ahead for PhD, or you don't want, like if you don't have a PhD right now, I don't think it is a problem. You need to collaborate to volunteer in research, like different research projects. Get the exposure out there. I mean, take the specialized certifications from from different good sources. You need to apply the bridge roles in the industry for now. If you don't have a PhD, like you can start with it with some analyst job, or a research assistant, or like a cheminformatics engineer maybe, or you can start with with the bio AI trainees and all of it. So yes.
Okay, okay. Thank you, ma'am, for the answer. Our next question is from Good Heart. If our college are not enough for research area, it's low facility college, then how can we do research in such area? Dr. Shubhi ma'am, any thoughts on this?
Yeah. Hi. So yeah, as you're asking, like it's not enough for the research area and slow facility. Yes, you can definitely do the research in such area. Okay, there is no restriction if your college does not have anything as such. Okay, nowadays we have like a lot of like the Google Cloud. We have like Google Colab, or we have like Jupyter. Okay, so we have all these platforms where we can do the research, where we have a lot of ample of space which has been given it to, like the given it, that will be given it to you, and there you can go, and then you can perform all your, like if you have like, if you want to perform like ML model, or you want to do the AI, okay, whatever kind of study you want to do, you can use those platforms, and you can work very well with these things. Okay, so there is no harm, like if there's nothing that if your college is not giving enough research area. Okay, so you can always be a strategic person, and you can find a space for yourself. Okay, so you have like a different place platforms which you can use it. Yeah, I hope I have given your answer.
Thank you. Thank you, Shubhi ma'am. Our next question is from Yashashwini. Um, Dr. Mani Verma, if you could throw some light on this. All the hosts, I think you meant speakers, are pointing to one thing. We need to be accurate while giving input. But how can we predict the ligand which suits the protein which gives the output?
We need to be accurate while giving input, but how can we predict the ligand which suits the protein which gives the output? The second part I don't understand, like what. So from my understanding, like what I understood is that uh, if you don't know the binding pose of the ligand on the protein, how can we get? I don't understand the second part of the question, but of course, if you don't know the binding pose of the ligand, how it binds to the target, better to be considering some reference ligand. We have to read some articles to understand how the binding pockets or evolution is changing with respect to the family of the proteins. So based on the such conservative from the sequence and structures, we understand the binding pocket, and with the information we can choose the ligand. Okay, how it's going to be binding at the pocket based on the family of the protein targets which has been published before. I
Okay.
hope I answered your question, but yeah, once you know like how it's going to be binding at the pocket, then we can take that data, then we can use this different form of data. Yeah, like I mentioned that the graphical data, the graph data. So like a lot of packages, I don't remember the package's name at this moment, but some packages which automatically converts your protein ligand structured to the form of data which is graph. So like series of nodes and edges it makes. So from this we can take it further.
Okay. Thank you, sir, for answering the question. Our next question is from Yash. After MSc Organic Chemistry, for what should we go actually? Cheminformatics or AI chemist drug discovery? Dr. Snigdha ma'am, any insights on this, please?
So Yash, after MSc Organic Chemistry, you can go both the ways, Cheminformatics and AI Chemistry. The main thing that you have to select your interest. First of all, you should have an interest because, of course, you are getting into the pipeline. I mean, you're applying there, and then you'll be there with that field. So first of all, it's your interest. Cheminformatics deals with, again, because it is informatics, it will deal with the databases, it will deal with the basics of the machine learning. AI chemist is more about, maybe, actually, these areas are same more or less. These are the same areas, just little bit of a difference, that the cheminformatics, maybe you'll be just entering into the roles like cheminformatics, and once you gain a lot of insights into machine learning and artificial intelligence, you will grow into the AI chemist area. So I think they are more or less the same thing. Cheminformatics is something to start with, and then AI chemist is like more of the, when you grow more, then you can start with AI chemist or enter into that league. I
Thank you,
hope that
ma'am.
answered your question. Yes.
Thank you, ma'am. So next question is from Veena Vani. I am MSc Organic Chemistry student. Can I become AI chemist? Dr. Shubhi ma'am, any insights, please?
Yeah, sure, ma'am. So, hi, Veena. So, as you are already a organic chemistry student, so you can definitely become an AI chemist. Okay, so you are already aware of the background of chemistry, right? So you're already aware of chemistry, what's happening, okay, what is there. So you just only have to learn a little bit more about the languages, like what I can say, like the different languages or the different tools which we have seen through the webinar. Okay, so you can just have to learn all these things, and you can definitely become an AI chemist because you are already from the chemistry background and you must have a good knowledge of the chemistry. So it is, in fact, much more easier than somebody who is coming from the non-chemistry background. So yes, you can, for sure, you can become a AI chemist, Veena.
Thank you, Shubhi ma'am. So our next question, so this is an interesting question from a LinkedIn user. A major challenge is the failure of drugs in late stage clinical trials. So this participant wants to know how would the future career formula, what we discussed today, aim to de-risk this process and improve translational success. Dr. Mani Verma, what are your thoughts on this particular question? If you could highlight some of your thoughts.
Yeah, that's true that like not all the drugs which has been generated by the AI would be effective at the end. But if we fine tune it based on the data that we are feeding to the model, of course, we can try to get the compound which is having the potency or the activity. If it is like 30%, we can take it as a hit. We can optimize it further. So not it need not to be necessarily a 0% or like 20%. Like even if it is a ligand with like least amount of activity, we can try to improve it at the stage when it gets failed. Yeah, so we can try to improve the structures by the classical way of drug discovery, such as ligand-based drug discovery, not only relying on AI. Okay, AI can give you like 10%, like 15% of it. So from that, we can take it further. So yeah, this is my answer.
Okay, thank you so much, sir. And Raghav Reddy has asked, I'm not a chemistry background, I'm from biotechnology. How is it helpful to me? Like, I need strong basics of chemistry to survive in industry level. Is it true? What do you think, Shubhi ma'am? Any insights on this? I'm not a chemistry background, I am from the biotechnology background. Or rather, any of the speakers, please, if you could help
Yeah,
out
so
the student. Yeah.
yeah, ma'am. So what I would suggest, no, Raghav, it's like you're as you are telling, you're from the biotechnology background, and it will be helpful. So I need to. No, Raghav. So what just happened over a period of time once you are suppose like in the industry or like once you start to learn the course, right, once you start like the bioinformatics, learning the basics of bioinformatics, and you go for the drug discovery field and the drug designing field, there you will be learning about like the basics of the chemistry, like the how the canonical formulas and every other thing will be okay. So once you start to learn this, I don't think so like you really need a very strong background of chemistry, but yeah, little bit you should. That's what I would say. Like over to the
Okay.
other speaker if they have any insight on this.
Okay. So we will pick up our last question now. So if Snigdha ma'am could answer this question, in our 30 days training, AI in Cheminformatics, will there be any recorded lessons or live lessons? And our whole concepts like RDKit, Cylon, QSAR, docking, and all will be covered in that. Um, Jay wants to know about it.
Hi Jay. It will be live sessions first of first. It will be like purely live session, and also one-on-one discussions. I mean you can put up your questions, you can put up your doubts in between, and then we'll be taking all the examples. We'll be showing you the demos also. We'll be having the hands-on sessions. We'll be having the do-along sessions also. And all the recorded sessions will also be uploaded. Right. I mean if at all you leave like you had not have a chance to to attend any lecture for any reason, you can see the recorded lectures as well. We'll be uploading all the recorded lectures with with the demo practice files, with the go along sessions, with assignments and everything. You can do those assignments, you can submit them, you can anytime mail all your like any one of us, like your experts, and then have any kind of discussions based on whatever your doubts are. And regarding your second phase of a question, that whether we'll be covering the RDKit scikit-learn QSAR docking, yes, we'll be covering the basics of chemistry, the basics of the informatics, what are the databases that you need to know, and then going ahead, what will be your ML pipelines, how you use RDKit, scikit-learn, TensorFlow, all of that. I mean, deep learning things, and then QSAR modeling, docking, simulation studies, a bit of it. I mean, what, like, so we are doing it all in all. I mean, it is highly, like, it is curated with the stepwise process, with with all the do along in demo processes. So I hope that answered your question.
Yes, that's right. So it is a complete comprehensive course that we have, and we are basically preparing you and we are making you future ready. That's what the webinar is also for. So with that, we come to an end of our Q&A session. Thank you once again to all the wonderful, for all those wonderful questions, and a heartfelt thank you to our speakers, Dr. Tiliban Manivarma, Dr. Snigdha Tiwari ma'am, and Dr. Shubhi Singh. Thank you so much for taking our time today and for sharing your expertise and inspiring us with your perspectives on how AI is shaping the future of pharma and biosciences. To all our attendees, remember this: every great scientific revolution begins with those who dare to learn something new before everyone else does. AI in Cheminformatics is that revolution. So don't wait for the future to arrive, be the one who creates it. So thank you for joining us today, and we look forward to seeing many of you here in our upcoming AIML and Cheminformatics batch, which starts 28th October. With that, we come to an end. Have a great evening ahead and keep innovating. Thank you, everyone.